Welcome 12th class students, Mrs. Nanda Biology tutorial. Today I will continue the previous chapter, that is the molecular basis of inheritance. First I have discussed up to the DNA structure. Now today I will discuss according to NCERT the topics continue chapter of molecular basis of inheritance. So next subtopic is transforming principle. In the previous we have discussed that DNA acts as a genetic material which carrying all the information from generation to generation. Now the experiment proved whether DNA is a genetic material, protein is a genetic material or RNA is a genetic material. But today the very important experiment 2, one is called to be Griffith experiment to prove transforming principle. That is, what is that principle? So he done the experiment with Streptococcus pneumoniae, that is, or Diplococcus also. They are causing pneumonia to the mice, and the experiment was done on mice. So he observed that two types of strains are available or present or causes the disease or not causes the disease. First one is called to be S strain. Second is called to be R strain. So what he did, Griffith experiment, he injected S strain and he found mice die. Second injected R strain, then he found mice survived. So S act as a virulent. Virulent means which causes disease, but it is disease that is a pneumonia. R is called to be non-virulent. So S virulent and R virulent, two strains of the bacteria, the culture. Then what he found? S mice die, R mice survived. Then third is S strain but heat. Heat killed S strain. Then he found mice survived. Next, S strain, which is heat killed, plus R. S strain heat killed plus R, then injected. Next, injected. So what he found, that is, now mice die. That mice die. What does it mean? Something was special, or something was very much important which transferred from one to the next. That means S strain heat killed to R strain, and R strain now become as a virulent which causes disease. So he proved that something is transferred from one to the next and mice die. So you can write this according to NCERT. This is the, you will get the full credit. Same, S die, not die, survived. So that is called to be the main 3 marks question. Strain. Even you can draw the diagram in certain book given as diagram, but be careful when mice die, then what? The mice should be in inverted position. That means in death, not in state 1. So book, one should be very, very confident if you are drawing the diagram of the mice. So that means die means that is inverted form. Ulta show karna hai aapko. So that is the experiment, and this experiment is very important and come for 3 marks in the paper. Griffith experiment. Explain Griffith experiment. This experiment done by Griffith on Streptococcus pneumoniae, which causes pneumonia. Then comes the second thing or second topic according to NCERT, that is called biochemical characterization or characterization of transferring principle. Second subtopic given according to the NCERT. So the number of the scientists, they discovered again which thing is special transferred from one to that. So number of the scientists, MacLeod, Avery, you can learn the name given in NCERT. MacLeod, Avery, McCarthy. So be careful of the spelling and the scientist's name. So they discovered what they have done, that they have taken the biochemicals of this particular thing. They proved that these are not proteins, they purified proteins. Purified proteins also. So their proteins are not responsible for these changes. So the certain material, that is a DNA, is responsible. Then they tested on their certain enzymes, DNAs, different proteases. Then they found that these are not responsible for transforming principle, and the subtopic is called biochemical characterization. So you can learn according to NCERT, right? So first, then comes the next experiment done by the scientists called Hershey and Chase. Very important experiment for board. Hershey and Chase. Hershey and Chase, the next experiment by Hershey. Hershey and Chase experiment, again on bacteriophages. What does it mean? Virus always take the name on which they attack. If you attack on bacteria, bacteriophage. If you attach on cyanobacteria, cyanophage. If on animals, zoophage. If on plants, phytophage. So number of the names of this given. So experiment done on bacteria that was taken, and they are radioactive. So bacteria. So cultures were made. One culture with S, radioactive S35 sulfur, and second P32. This culture was made, and this is Hershey and Chase experiment. So this again important according to NCERT. They are very important. One, what 3 steps you have to write for the paper. One is called to be infection, second is called to be blending, and third is called to be centrifusion. So 3 parts. Culture was made, and after that bacteria. Suppose this is a bacteria. Then this is the virus structure, as you know. Virus. In this case, they effected on bacteria. This is called to be sulfur 35 and phosphorus 32. In one case, this is the protein code and this is the DNA. In one case, DNA is radioactive or labeled. In second case, protein code was labeled. So infection was done. Then blending. That means blending and centrifusion. After that, there is a formation called supernatant and pellets. Then it was found 3 steps you have to write: infection, very important, blending, and centrifusion. Right? Infection and centrifugation, or FUGATION, centrifugate, like this. So these are the diagram. Even you can draw these diagram, you will get the full credit for the board. So see the cases. What is this diagram? In this case, it is affected by the virus, bacteriophage. In one, DNA. In second, protein code. So after infection, what happened? The protein code remain outside and the genetic material genetic material which enters into the bacteria start replicating along with that. So it means it was found the labeled or radioactive S35 found labeled new viruses or small detected but no radioactive in the second case P32. So it proves that the genetic material DNA was labeled, transferred from this virus to inside and start replicating along with it. But in the second case, protein coat which remain outside, left outside, same protein coat left in both the cases outside. So DNA enter, like this, DNA enter. So it start replicating along with this bacteria. So it proves that DNA act as a genetic material, and this experiment is called Hershey and Chase experiment. Very, very important for board for 3 marks. So diagram same you can draw of your NCERT, and 3 word, don't forget infection, blending and centrifugation. So that means DNA. So 2 experiments are very important. One is Griffith experiment, second is Hershey and Chase experiment to prove DNA as a genetic material. Third subtopic is called that what are the properties of DNA? Third step according to NCERT, properties. What is the properties of DNA? PRO, properties of DNA or genetic material. That genetic material should be more stable. First point. Next, they can give the replica. Next, they show mutations, but at slow rate mutation, replica which take part in evolution also, they are more stable, right? Less reactive. So it means properties. So these are the properties are present in DNA. RNA also having this, but they are unstable. They are more reactive than DNA. DNA is a double-stranded, but RNA is a single-stranded. They mutate very fast, but DNA mutate at slow rate. They show the mutation, but at slow rate. The mutation are the sudden changes. Now you know that this virus, now these days, that is a coronavirus. So that means they mutate and can change the genetic code very easily. So this is the property of a genetic material. So it proves that DNA is more stable. Similarly, DNA having thymine in the structure, they are having thymine which is more stable than uracil. So they are having very important properties. So it means DNA acts as a genetic material. Then comes another, according to NCERT subtopic, DNA replicate or replication of DNA. Replicate, divide, give the replica or carbon copy. So DNA replicate, yes, but very important, they are semi-conservative in nature. Suppose this is the DNA. So DNA show the replicon or one point where they replicate. Prokaryotes having single replicon, it is called replicon. Replicon, where they replicate, they divide. So when they divide, one strand remain as parental and second strand form new. It means they divide here like this. One is parental, one is totally parental, second is new. Same, divide into 2. So this nature is semi-conservative. That means they divide, but one strand remain as parental and second strand remain or they form new one. This is called the semi-conservative, and question come from the board for 2 marks. What do you mean by semi-conservative nature of the DNA. Then what is the main process of replication? They form the replication fork. In NCERT, there is a written replication fork. What does it mean? This is a DNA. The point, replicon or ori, origin of replication, ori center, which give the new stand. So the various steps of first is activation of deoxyribonucleic acid. So activation of deoxyribonucleotides. It means they are freely present in the cytoplasm. They occur in the form of DAMP, DCMP, right? So this is called, MP means monophosphate, adenine monophosphate, thymine monophosphate, TMP, GMP. So deoxyribonucleics freely. So first step of the replication is they are present freely in the cytoplasm. Then they are phosphorylated. Phosphorylated means they combine with IP, inorganic phosphate. So AMP, right? GMP, TM. So they changes into ATP formation, monophosphate, TP, CTP. So that means they are in the triphosphate. Monophosphate changes into triphos. And this is the first step called activation of deoxyribonucleotide. Activation of deoxyribonucleotide. So it means after the activation, then DNA strand exposure of parental strand. There is exposure. This is the origin of replicon, where they break and they expose. And arrangement, as we know, they are double stranded structure, 3:5 or 5:3. 3:5. So replication, that means they replicate and they form fork. For 2 marks question, replication fork means exposure of the parental strand. So what happened? Then they need on this side RNA primer or RNA primase. They form without any gap continuous strand, RNA primer. Similarly, 2 enzymes which are responsible for unwinding and breaking this or separating, they are called topoisomerase. This is called helicase. So then after that exposure, RNA primer continues. First comes exposure of strand, then exposure of the parental strand, then there is a formation of RNA primer which is act as a starting point. Then the complete strand is formed, which is called to be leading strand. Complete strand is called to be leading strand, continuous strand, leading. So it means this is a complete strand, leading strand. On the second side, the strand or the structures which are formed, they are formed in fragments. They are called in fragments, and these fragments are called Okazaki after discovery of the scientists. Okazaki fragments. So they are in this side, but they are not continuous. Here is the continuous. So it means every fragment has to be formed by the action of new RNA formation. Here they need only one RNA starting. This gap or this strand then become continuous by the action of the next enzyme called DNA ligase. DNA ligase to join. So this is the point of the replicon. exposure. So after that, then they separate from the parent. One strand is new, another is parental. This nature is called to be semi-conservative nature of the DNA. Then come Okazaki fragment. So it means first step is called activation of deoxyribonuclei. Second is called to be exposure of this strand or DNA strand. Third is called the formation or starter point, that is called to be RNA primer or primase. Then comes new strands are formed and they undergo next phenomena called base pairing. Means here is a adenine, then automatically thymine, cytosine, guanine, the pairing occur. Double bond and triple bond we discussed in the previous one. You have first learned the structure of DNA. So that means the base pairing occur. By mistake, if the base pairing is incorrect, not matching, then this DNA strand go back and bring their own. This is the quality, this is the capacity, this is the property of DNA. They can bring their own specific base pairing. So it means that is called to be proofreading. You know, which you write something and after that you do proofreading. Whether it is right or wrong, same the DNA does. And this phenomena is called to base pairing. It means activation. First deoxy, second exposure, third base pairing, then finally leading lagging strand. That means the joining. After that there is a proofreading by the DNA. Together constitute the method and main property or how the replication fork is formed. This is called replication fork or replication of DNA to form replication fork, which is very again important. In the board it comes like this. This is the stand. Sometimes they give only the stand and you have to label the various parts. They give the arrow and give which one is parental, which is new synthesized, which is the old stand. So that is replication fork formation. So it means semi-conservative replication. That is another property or quality of the DNA which act as a genetic material. So up to here I think it is clear to you. The second video. Thank you.